After the DNA has been replicated, the last thing that we need to do is to package the new DNA. The details of DNA packaging are a little bit different between prokaryotes and eukaryotes. Prokaryotes have one circular chromosome. Therefore, the replication of prokaryotic cells is pretty straightforward. First, we're going to have the origin of replication. This is a somewhat short region of DNA, about 250 base pairs long, that has a lot of A's and T's in this origin of replication sequence. Why do you think that the origin of replication would have more AT pairings than GC pairings? Hey, that looks like a good essay question on like an exam or something. So from that origin of replication, we are going to see replication occurring in both directions on that circular chromosome. When that polymerase gets around and they meet up on the other side, that's when we know that DNA replication is done. We then have two daughter DNA strands that are ready to go into the new cell. Prokaryotic cells use supercoiling and specialized proteins to wind up their one circular chromosome into that nucleoid region. So if you've ever had a hairband, right, one circular chromosome, and we can twist that up into a really tight knot. That's kind of what we're doing here. Eukaryotic cells have much larger genomes. For example, E. coli, which is a bacterium, has a genome about 4.6 million base pairs long. Humans are about 6 billion with a B. And ferns, some species of ferns, can have extremely large genomes. Additionally, eukaryotes have multiple linear chromosomes. So instead of one circular chromosome like we see in prokaryotes, we have these large, long chromosomes. And to copy all of this DNA efficiently, eukaryotes will have multiple origins of replication along a chromosome. So we're going to have a lot of starting points. Once the DNA is replicated, it has to be packaged. The DNA double helix first wrapped around special proteins called histones. The histone protein plus the DNA is called a nucleosome. And those nucleosomes are going to be coiled to form chromatin. When it's time for the cell to divide, the DNA is going to condense further into chromosomes. When talking about the compaction and the packaging of DNA in eukaryotes, I like to talk about this set of china. My mom purchased this set of china when I was a baby, piece by piece. And when I became an adult, she asked if I wanted it, and I was like, yeah, I want that. And most of the time, this set of china doesn't get set out. These are not our everyday plates, right? These are special. These are irreplaceable. There's, surely 40 something years later, you cannot purchase these pieces. So if this bowl gets broken, then that's it. Let's pretend that they live in a china cupboard. But when I moved to Arizona, I wanted to make sure that these pieces were very well protected during the move and that they wouldn't get chipped or broken during that process. So they got packed up in this box they're clearly labeled. Each piece is wrapped in bubble wrap, and all the spaces in between are packed tightly with newspaper and other, scraps of bubble wrap so as to protect these. And that's kind of what the DNA is. So when it's time for Thanksgiving dinner, we'll pull these out and use them
but when they're not currently being used, they get an extra layer of protection. And when it's time to move longer distances, that's when they get the highest degree of protection.
Linear chromosomes that we see in eukaryotic cells creates a problem. The ends cannot fully be replicated, causing the chromosomes to become shorter and shorter every time. And remember that the cells in your stomach lining are replaced every 2 to 3 days. So this shortening, because of the lack of ability to copy that, can mean that over time, the more the chromosomes are copied, we're going to start losing information. Telomeres are act as protective chromosome caps or margins. During the Middle Ages in Europe, books were all written by hand, which, as you can imagine, is incredibly time consuming. If the scribes wrote all the way to the edges of the paper, their work had the potential to be damaged by water, dust, rodents, and other aspects of medieval life. So they kept about an inch space between the edge of the page and the edge of their writing. This protected the information on the book from damages from the outside. Also, margins are a great place for them to draw totally unhinged doodles. Telomeres are kind of like the margins of those medieval texts. They protect the important information from getting damaged. So when a cell divides, the DNA that can't be copied is nonsense anyway. A former student of mine said that the telomere sequences are like the lorem ipsum text. It's filler words that don't really mean anything anyway. Telomerases are enzymes that extend the telomeres. So here's our telomere. The telomerase is going to extend that sequence and reduce the chromosomal shortening. Because of this role, telomerase is heavily studied in aging and cancer research.